Welcome to this explainer on the principles of inheritance and variation. Think for a moment about the absolute most complex piece of machinery you've ever laid eyes on. Got it? Okay, now realize that the instruction manual for building something infinitely more complex, which is you, is literally packed into almost every single cell of your body. Today, we're going to decode that biological instruction manual and explore the absolutely fascinating world of genetics.
It's a question we've literally asked for centuries, right? Why do you look like your parents, but, not exactly like them? Well, the answer lies in two core concepts. First up, inheritance. That's the process where characters are passed down from parent to progeny. And second, variation, which is the degree to which that progeny actually differs from their parents. These two forces kind of dance together to create the incredible diversity of life we see all around us.
Here is our roadmap for today. We'll be covering 1. Mendel and pea plants, 2. Beyond simple dominance, 3. Chromosomes and sex determination, 4. Tracing traits through pedigree analysis, and finally, 5. Mutations and genetic disorders. Let's dive right in.
All right, Section 1, Mendel and pea plants. Time to look at the foundational rules of inheritance. To really grasp this genetic language, we have to start with Gregor Mendel. The guy spent 7 long years, from 1856 to 1863, just studying garden peas. Now, to understand his work, we need a little vocab. Think of genes as discrete units of inheritance, and alleles as, well, there's slightly different variations. The hidden genetic code, say 2 tall alleles, TT, or 2 dwarf alleles, TT, that's your genotype. But the physical trait we actually see, like whether the plant is tall or dwarf, that's the phenotype. If the 2 alleles are identical, making a purebred trait, it's homozygous. If they're different, it's heterozygous. Make sense?
So, when Mendel crossed a pure tall plant with a pure dwarf plant, the entire first generation was tall. The dwarf trait just seemed to vanish. But, and here's where it gets really interesting, when he let those hybrids self-pollinate, the dwarf trait mysteriously reappeared in the next generation. Out of all the offspring, 75% show the dominant tall trait and 25% show the recessive dwarf trait, a perfect 3-to-1 ratio. This brilliantly illustrates Mendel's law of segregation. Alleles don't just blend together, they separate during gamete formation, meaning a recessive trait can hide out completely undetected for a whole generation before making a sudden comeback.
Moving on to Section 2, Beyond Simple Dominance. Because Mendel's strict rules, they get a little more complicated.
So we gotta ask, does one trait always strictly dominate another? Mendel definitely thought so with his peas, but as scientists kept digging, they found some really fascinating exceptions where the rules get kind of blurry. Biology is rarely just black and white.
Sometimes traits just blend. Look at incomplete dominance. If you cross a red-flowered snapdragon with a white-flowered snapdragon, the offspring don't look like either parent. They're pink. The alleles literally mix together. But other times, they completely share the stage. That's codominance. A perfect example is human ABO blood types. If you inherit the A allele from one parent and the B allele from the other, neither one dominates. They both actively produce their specific sugars on your red blood cells, giving you type AB blood.
Section 3, Chromosomes and Sex Determination. Let's look at the actual chapters of our instruction manual.
It took decades to piece all this together. Mendel published his work in 1865, but honestly it went largely unrecognized. Then in 1891, a scientist named Henking observed this mysterious structure in insect sperm that he simply called the X-body. It wasn't until 1900 that 3 independent scientists finally rediscovered Mendel's work, and shortly after, linked his principles with the physical movement of chromosomes. That cracked the code, sparking a golden era of discovery that eventually led to Watson and Crick winning the Nobel Prize in 1962 for solving the double helix structure of DNA itself. Okay, let's dive into some wild variety here. It is fascinating how different species determine sex. In humans and fruit flies, we use the XY system. Males are XY, females are XX. But look at grasshoppers. They use the XO type. Males literally just have one X chromosome. No second sex chromosome at all. And then, the honeybees. They use a haplodiploid system. Females develop from a fertilized egg, but males, they develop from an unfertilized egg. That means a male honeybee has half the chromosomes of a female. He literally has no father, he cannot have sons, but he does have a grandfather and can have grandsons. Nature is just wild.
Which brings us to Section 4, Tracing Traits with Pedigree Analysis.
Obviously, we can't easily perform controlled genetic crosses in humans. We take way too long to grow up. Instead, scientists use pedigree analysis. This is basically the study of family history regarding how a particular trait is inherited across several generations. It's essentially a detective's tool for mapping the human genetic manual, letting us trace exactly how a specific gene travels down a family tree. To read a pedigree, you just have to crack the visual cipher. It's pretty straightforward. Squares represent males, and circles represent females. If a symbol is filled in or shaded, that person is affected by the specific trait or disease we're looking at. Unfilled symbols mean they're unaffected. A single horizontal line between a square and a circle indicates mating, while a double line indicates mating between relatives. By mapping all this out, geneticists can figure out if a disease is dominant or recessive, and whether it's linked to a standard autosome or a sex chromosome.
Finally, Section 5, Mutations and Genetic Disorders. What happens when there are typos in the code?
So what actually happens when there are typos in this biological instruction manual? Well, these typos are called mutations, alterations in DNA sequences that literally change an organism's genotype and phenotype. They can be triggered by mutagens like UV radiation. You can have deletions or insertions of DNA segments which cause massive, highly disruptive frameshift mutations. Or you might just have a point mutation where literally just a single base pair of DNA changes.
When these mutations cause disease, we generally group them into two main categories. Mendelian disorders are caused by a mutation in a single gene. Think of it as a single misspelled word in the manual. Examples include cystic fibrosis or hemophilia. Chromosomal disorders, on the other hand, happen because of an absence, excess, or abnormal arrangement of entire chromosomes. This isn't just a misspelled word. This is an entire page of the manual missing or duplicated, like in Down syndrome.
I want to throw a fascinating number at you regarding Mendelian disorders. 8. That is the percentage of males who are colorblind. Now compare that to a tiny 0.4% of females. Why such a massive difference? Well, colorblindness is a sex-linked recessive disorder found on the X chromosome. Since males only have one X chromosome, a single mutated gene guarantees they're going to be colorblind. Females have two X chromosomes, so they need the mutation on both to actually have the disorder. Otherwise, they just carry it hidden, kind of like how Queen Victoria famously carried hemophilia.
So, the crucial point is understanding how the nature of the typo defines the disease. Let's compare two severe blood disorders. Sickle cell anemia is a qualitative problem. A point mutation substitutes just one single amino acid. The hemoglobin is there, but its quality is completely ruined, making the red blood cells painfully sickle. Thalassemia, on the other hand, is a quantitative problem. The body simply produces way too few of the required globin genes.
And then chromosomal disorders really show the dramatic effects of aneuploidy. That's the gain or loss of an entire chromosome. Down syndrome is caused by an extra copy of chromosome 21. Klinefelter syndrome happens when a male has an extra X chromosome, 47XXY, resulting in overall masculine development but with some feminine features. And Turner syndrome is the absence of an X chromosome in females, which results in sterility. Just think about that. Gaining or losing just one chromosome out of 46 completely alters human development. From Mendel counting garden peas in the 1800s to us mapping the exact molecular typos that cause disease today, our understanding of inheritance and variation has just exploded. Which brings us to a final provocative thought. If we can now accurately read the typos in our genetic instruction manual, how long until we start rewriting the book entirely? Thank you for exploring the blueprints of life with me on this explainer. Keep wondering and keep learning.